This is an oral presentation for the recombinant expression of superoxide dismutase SOD1 using a vector in a competent E. coli cell. The SOD1 is an antioxidant protein found in the mitochondria and cytoplasm of aerobic cells. It functions as a scavenger of superoxide radicals, free radicals, by converting them to molecular oxygen and hydrogen peroxide, which are not harmful to the cell. Now, some studies have been conducted that the SOD1, the misfolding of the SOD1 and the toxic conformation of this protein leads to a disease known as amyotrophic lateral sclerosis. And so it is my wish to express this protein in order to study it and possibly identify a potential drug to treat this disease. First of all, to go about this, we need to get a suitable vector. In this case, we're using the PET22B vector cell, or vector or plasmid, which is a vehicle to carry our gene of interest, the SOD1. So what happens? This vector has the multiple cloning sites. It has a T7 lac promoter for expression of protein, because it's a very powerful tool for protein expression. And then it has the terminator flanking the multiple cloning site. It also has the N-terminal sequence as well as the C-terminal sequence with the with the polyhistidine tag. PET is a widely used vector for recombinant protein expression. So now what happens here is that we use the restriction enzyme NDE1 and SAL1, which are type 2 restriction enzyme, to digest our plasmid, our vector, and then at the cloning site, and then we'll do the same thing for our gene of interest, the SOD gene, and then introduce this gene into the plasmid using the T4 DNA ligase. Now, this is the ligation process, or the ligation. The ligation now takes place. So what then happens is that we take our recombinant DNA, or the chimeric plasmid, because it contains the eukaryotic gene and prokaryotic DNA. We now incubate this the solution of the cells on ice for about 10 to 30 minutes. Now this is to allow for proper ligation to take place. And then we will now transform the E. coli. Usually the transformation of the E. coli is introduction of the recombinant DNA into the E. coli. So this is done by the heat shock. Our solution is then placed on a water bath at 42 degrees Celsius for 45 seconds. After which we quickly take it off the water bath and then place on ice for about 2 minutes. And then we add our SOC or any other recovery medium that allows or improves molecular uptake while stabilizing the cells. This also allows the bacterial strains to pick up the recombinant plasmid. Then we incubate our culture at 37 degrees in an incubator that is shaking for about 45 minutes to 1 hour thereabouts. Before we now transfer this bacteria on an antibiotic plate, LD medium. That's a LD medium containing ampicillin. Now remember that our vector has the ampicillin resistant gene. So what happens is that the bacteria placed on the antibiotic plates will now distinguish between bacteria that has the ampicillin resistant gene and those that doesn't have. Those that doesn't have the gene will die off, while the ones that has the ampicillin resistant gene will now propagate. They will now grow and form colony. So we now introduce IPGT to give a good yield of the desired protein, which is our SOD1 in this case. Then we'll carry out the extraction or the lysis of this cell and of this cell. Later on, we now, because our vector has the histag sequence, we now use it um, to bind to the histag now binds to our protein of interest, which will then be purified using using affinity chromatography. We can also use the Western blot analysis using 15% SDS polyacrylamide gel electrophoresis and the PVD membrane. Even we can also try using the Sanger deoxy sequencing to purify our protein. This is what happens in this experiment.